Hello everyone, and welcome to today's webinar, Use of a New 3D Pluripotent Stem Cell Suspension Culture Medium for Rapid Scale-Up and Downstream Differentiation of Pluripotent Stem Cells. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com/StemScale. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This presentation is educational and thus offers continuing education credits. Please follow the process to obtain your credits by clicking on the Continuing Education Credits window. I'd like to now welcome our speakers. Michael Akenhead, PhD, Scientist 3, Biosciences Division at Thermo Fisher Scientific, and Michael Derr, Scientist 3, Biosciences Division at Thermo Fisher Scientific. Dr. Michael Akenhead will be present for the first half of this presentation, followed by his colleague, Michael Derr. Dr. Akenhead, you may now begin your presentation.
Thank you for the introduction. So I will be starting by going over how we're using our new 3D pluripotent stem cell suspension culture medium called StemScale for the rapid scale-up and downstream differentiation of pluripotent stem cells. So here's a brief overview of my part of the presentation. First, we'll be covering briefly the benefits of suspension culture. Then we'll be discussing the Gibco stem cell PSC suspension medium workflow. And then lastly, I'll be focusing on some StemScale performance data. So first, let's talk about why researchers want to utilize suspension culture. Adherent cultures are limited by their surface area and also by the confluency of the cells grown in that area. Whereas in contrast, suspension cultures are limited by volume instead of surface area. So this enables a higher capacity for growth and suspension due to increased efficiency and higher seeding densities. So overall, the main appeal of suspension cultures is the ability to more rapidly scale up to large numbers of cells. This can be accomplished in a shorter period of time, as you can see in the figure at the bottom, calculated 19 days to achieve 4.5 billion cells in suspension, compared to adherent cultures, which in this case show 28 days to achieve the same 4.5 billion cells. In addition, suspension culture is also less expensive than adherent culture due to suspension cultures utilizing less plastics and consuming less media in comparison to adherent cultures. Although suspension cultures are very appealing due to the advantages they have over adherent cultures, there are still some technical challenges which must be overcome. And this can be observed when comparing two main categories here, microcarrier cultures versus spheroid cultures. So the microcarrier cultures involve cells growing on some sort of supported matrix, such as beads, gels, or other structures. And while this does enable consistent cell growth, they tend to be time-consuming to prepare and difficult to passage. There's also inconsistent attachment of cells to the microcarriers. And overall, the limited surface area on the microcarriers can sometimes prohibit the large-scale expansion of cells in suspension. So in contrast to the microcarriers, spheroid cultures are much more appealing to researchers. These cultures utilize the ability of cells to self-aggregate into floating spheroidal clumps, thereby eliminating the need for cells to grow adhered to a microcarrier substrate. And while this does simplify the initiation of suspension cultures, there are still some drawbacks to the spheroid culture, particularly related to current solutions. In particular, it's difficult to regulate the aggregate size and growth across multiple different PSC lines in order to achieve consistency. Additionally, some current solutions have a relatively low fold expansion in cell number, making scaling up to large numbers more difficult. And lastly, oftentimes there are cumbersome workflows involved with spheroid cultures, requiring the use of cell strainers or other components that make it more difficult to easily passage. So when we took all these factors into account, we wanted to come up with a suspension culture medium that can maximize the expansion capability of cells and simplify the workflow while overall maintaining their pluripotency. So to accomplish this, we came up with the StemScale suspension medium, and we focused on these main components when designing StemScale. We wanted to make expansion efficient, thereby optimizing spheroid nucleation in order to promote efficient growth. We also wanted to simplify the workflow. So to do this, we focused on enabling periodic medium exchanges and focusing on the ability to passage cells without the need for extra components like cell strainers. We also wanted to have consistent scalable cultures across multiple size vessels, and also the ability to maintain cells pluripotent when they're grown in spheroids. And lastly, we wanted to generate high quality PSCs capable of being used for downstream applications such as differentiation protocols. So now we'll briefly discuss the StemScale workflow here. So here's the StemScale workflow at a glance. This workflow involves seeding of 3D culture vessels on day 0. And after seeding, we utilize a 50% medium exchange in order to feed cultures every other day. This replacement strategy prevents the accumulation of waste in the suspension culture and also contributes to the maintenance of a consistent spheroid size, since spheroids will be grown in a constant volume for the duration of the culture. And as I mentioned, the 50% medium replacement strategy illustrated here is an every other day feed, although you can feed cells daily in order to maximize their viability. So in the example here for an every other day feed, we are skipping feeding on day 2 and day 4 in order to passage cells on day 5. This is a typical 5-day passaging schedule, but you can passage cells on day 4 as well, or even as early as day 3 if you want to avoid passaging on the weekend. So in addition to StemScale medium, there are some other components that are necessary to have as well. It's also important to have an orbital shaker platform that can be placed into a cell incubator, the StemPro Accutase passaging reagent, and the desired vessel which will be used to grow suspension cultures. So for the desired vessel, we recommend non-tissue culture treated plates if you're using well plates, or a plain bottom flask that is non-baffled if you're using some sort of shake flask. So with all these materials in place, you're ready to initiate these StemScale cultures. These cultures can easily be seeded from existing adherent cultures. And once your adherent cultures are around 60 to 80% confluent, then you can dissociate them using the StemPro Accutase passaging reagent and then seed them into your desired vessel of choice. The recommended seeding density is 150,000 cells per mil, although you can seed in a lower density. And when seeding cells, it's also important to remember to include a ROCK inhibitor. We recommend using Y27632, but other ROCK inhibitors are possible as well. Without the ROCK inhibitor present, you'll find that the cells will not spontaneously aggregate together into these spheroidal clumps. So after seeding the suspension cultures on day 0, the cultures will need to be fed on the subsequent day. And because these suspension cultures have formed spheroids overnight, we can use gravity sedimentation to collect spheroids at the bottom of the well or the flask, thereby enabling medium exchange from the top of the vessel. The gravity sedimentation should occur for approximately 5 minutes in order to minimize the amount of time the spheroids have been clumped at the bottom of the well. Because if spheroids remain sedimented for too long, then they will begin to aggregate to each other and form these undesirable clumps. This can also occur if the spheroids are removed from agitation for an extended period of time, such as when you're imaging spheroids under a microscope. So it's important to try to minimize the amount of time that plates or flasks are removed from an orbital shaker platform. So no longer than 15 minutes is a good recommendation. So to accomplish performing the 50% medium replacement for the well plates, you can tilt the well plate at a 45-degree angle and then wait 5 minutes for the spheroids to settle. For the shake flasks, you can simply remove this flask from the orbital shaker platform and then just let the spheroids fall to the center of the vessel. Then after 5 minutes, you simply remove half of the medium, the spent medium that is, and then replace with an equal volume of fresh medium. Once this is done, you can just move the plate or the flask back onto the shake platform. So overall, the 50% medium strategy is very beneficial for suspension cultures because it's very simple to perform, only relying on the gravity sedimentation of the 3D spheroids. Additionally, because the 50% medium replacement strategy has an equal volume of fresh medium every time you're removing the spent medium, this ensures the culture volume remains constant for the duration of the suspension culture. And because spheroid size is affected by the shear force applied to the system, by maintaining a constant volume, you're ensuring that the spheroids are always being suspended and exposed to the same shear, thereby ensuring that their size remains consistent over the duration of the culture. And most importantly, the 50% medium replacement strategy will prevent the accumulation of waste products over the duration of the culture. Other types of feed strategies, such as batch feeding processes, which just add fresh medium to the existing spent medium, we'll see metabolic waste products accumulate far more rapidly compared to this replacement strategy. So in order to passage the StemScale cultures, we utilize StemPro Accutase. This is also a very simple process. Once the spheroids are ready to be passaged, they're transferred into a conical tube. We then centrifuge the conical tube in order to aspirate the supernatant, and then add the StemScale passaging reagent for about 10 minutes. Over this 10-minute duration, you can see the spheroids begin to break apart as gentle agitation is applied to the tube. So after the duration of the 10 minutes, the spheroids should be completely broken apart into very tiny chunks, making them nearly invisible to the eye. So then you can place them in the centrifuge, spin them down, and then prepare them for counting. And after you've obtained your cell counts, then you can use them for subsequent downstream applications, or reseed them back into suspension vessels for subsequent passages. So this passaging protocol has several benefits to researchers looking for a simple protocol. Most importantly, there's no need to utilize cell strainers or microcarriers in this passaging protocol. Cell strainers tend to be cumbersome to use, can often damage cells, and you can often even lose cells as you're passing them through the cell strainer. Additionally, since we're simply using Accutase with no cell strainer, there's limited manipulation that's required, reducing the risk of contamination. And there are also a few additional experimental variables which can be used to manipulate and control spheroid size and growth. So most important is the shaker platform RPM. As you see here, shaker platform RPM is inversely related to spheroid size. So by increasing the platform RPM, you'll have smaller spheroids as a result. The images on the right show spheroids on day 1 at 70 RPM compared to spheroids on day 1 at 90 RPM. And you can see at 90 RPM, the spheroids are much smaller compared to at 70. In a similar fashion, as I mentioned how the shear force is important to the spheroid size on the previous slides, culture vessel volume will also affect the spheroid size, and it's directly related. So at 30 mils of media in the shake flask, the spheroids will be smaller than grown at 40 mils of media. Lastly, seeding density is another variable. This one, by increasing the seeding density, you'll obtain a larger number of spheroids, or a greater number of spheroids in your suspension culture. But it's important not to increase seeding density too high, as with more spheroids forming and expanding in suspension, the media will also be consumed at a much more rapid pace. So now that we've discussed the StemScale protocol, I would like to demonstrate its performance with some experimental data here. So on this slide here, we see that StemScale promotes the rapid expansion of cells in suspension. So you can see a representative image of our Gibco-somal iPSCs in the upper left, and this is how the spheroids will appear after 5 days of growth to the StemScale medium. The two graphs shown here represent their growth in either well plates or larger vessels of varying sizes. And you can see that we obtain an average of approximately 10-fold expansion, whether they're grown in small well plates, larger shake flasks, or even larger bioreactors. So this indicates that StemScale does promote expansion and consistent expansion across a variety of different vessel sizes. And in addition to high expansion potential, we see that StemScale also maintains pluripotent cells over multiple passages. So on this slide, I'm showing that Gibco-somal iPSCs and WAO9 ESCs, which are H9 cells, exhibit over 90% expression of octoform nanog pluripotency markers as assessed after 5 passages in suspension. We further confirm this through the use of our Pluritest assay, indicating that cells from our 3D spheroids all remain pluripotent and distinct from non-iPSC controls. And we also confirmed that these cells obtained after 5 passages will maintain a normal karyotype as assessed through the karyostat assay. There were no chromosomal aberrations detected after 5 passages in suspension. So together, this shows that StemScale can both enhance fold change and maintain the pluripotency of cells grown as spheroids. Now, this slide I had shown for 5 passages, but we also have data over multiple consecutive passages for Gibco episomal iPSCs. And in this case, we grew them out 30 consecutive passages in suspension and then did the same performance tests. And so as you can see, the graph on the upper left, after 30 passages in suspension, they have the same over 90% expression of OCT4 and nanog markers. They also passed the Pluritest assay after passage 30, and again showed no chromosomal aberrations as determined by the karyostat assay. Although I don't have the data shown here, we also ran the cells on the Akenhead assay to look for specific oncogenic mutations, and saw that none were detected. And lastly, the graph on the bottom right shows that the cell line will maintain consistent growth over 30 consecutive passages, as we have a nice straight line with each data point representing 1 passage, indicating that we get approximately 8 to 10-fold expansion per passage. So in order to prevent the formation of a necrotic core when growing cells as spheroids, it's important to passage spheroids when the average diameter reaches a certain size. We found that passaging spheroids when the average diameter is approximately 400 microns will prevent the formation of this necrotic core. So when you're growing cells as spheroids in our StemScale medium, we found that this can occur on either day 4 or day 5 of culture. On day 4, the average diameter is less than 400 microns, while on day 5, it's right at or slightly above 400 microns. So users can choose to passage either day 4 or day 5, depending on their needs for cell yields. You can also choose to passage on day 3 if necessary, such as if day 3 is falling on a Friday and you'd rather not passage on a Saturday or a Sunday. But if you're passaging on day 3, the cell yield will be lower due to the limited expansion time that these cells have had compared to a day 4 or day 5 culture. Now, all the previous data that I've shown has been utilizing Gibco episomal iPSC cells, or H9, the WAO9 cells is listed. But I like to show that StemScale is compatible with a variety of different cell lines as well. So on this slide, I've got a comparison between 4 cell lines, including the previously mentioned ones, also WTC11 iPSCs and WAO1 ESCs, which are H1 cells. And although I don't have the data displayed here, when we were doing alpha testing with our media, our alpha testers also evaluated with their own in-house lines. So we've evaluated a total of 10 different cell lines as well. So if you look on the graph on the bottom left, you can see the expansion potential of all 4 cell lines. All of them are showing consistent growth over 3 consecutive passages. But what I want to point out is that the fold change, the expansion potential of each cell line in suspension will be different, depending on how easily the cells form spheroids. So for the Gibco episomal iPSCs and the WTC11 cells, they tend to form spheroids easily, so they have a much higher fold change compared to the WAO9 or the WAO1 cells, which have more difficulty nucleating and forming spheroids, and so they will have a lower fold change as a result. So on this slide, I'd like to show how StemScale performs compared to some other media that are currently available that are marketed as suspension culture media. So we evaluated our StemScale media against another type of media, and you see the cells on the upper left. Our StemScale spheroids tend to be larger than the spheroids obtained from the other medium. And when you look at the resulting fold change over 3 consecutive passages, we see that for our Gibco episomal iPSCs, we're averaging approximately 11-fold expansion per passage, whereas using the other medium, our Gibcos will only average approximately 5-fold expansion per passage. The graph on the upper right shows the OCT4 and nanog expression for both media, and we see that it's the same, greater than 90% expression. So this indicates that our StemScale medium is capable of outperforming in terms of growth of the other medium while still maintaining pluripotent cells over the course of these 3 consecutive passages. And so while I've been showing how StemScale is optimized for suspension culture and mentioning how it enables you to scale up to large numbers of cells, and in addition showing how it outperforms the other media on the previous slide, I do want to emphasize that this is not because StemScale is causing cells to divide more rapidly than what is observed in adherent cultures. When you compare the growth rates of adherent cells grown in StemFlex, the bottom left graph against spheroids that are grown in StemScale, we see that their population doubling times are similar. So this indicates that the rates of expansion are equivalent, and it's actually the benefits of the suspension culture system which support the ability to generate large numbers of cells. So when we optimized StemScale, we optimized its ability to nucleate and to efficiently grow spheroids to ensure we can take advantage of the suspension culture benefits. As I mentioned in the introduction, because adherent cultures are limited by surface area and by a rigid passaging schedule dictated by confluency, they're unable to achieve these large numbers of cells. Whereas suspension cultures are limited by volume, and with this extra dimension, they can support higher numbers of cells to be fed into cultures. And because spheroid size is what regulates the passaging schedule, this is much more flexible than confluency. It can allow cultures to grow for longer periods of time, thereby ultimately enable a rapid scaling up using the same population doubling time. And since I've been talking about being able to scale up to large volumes, on this slide I want to show that we've been able to scale up to a 3-liter bioreactor system using StemScale media. So the images on the left are from spheroids used in 125 ml shake flasks, small 100 ml bioreactor, and then the large 3-liter bioreactor. And the graph in the middle shows the fold change results that we obtained. And we achieved about an 11-fold expansion in the 3-liter bioreactor with our Gibco somal iPSCs, which is very similar for what we've seen on all the previous slides. I do want to point out that the 2 bar graphs, or the 2 bars for the 100 ml bioreactor and the 125 ml shake flask are a little lower than the 3-liter bioreactor. This is because I was sampling daily from all these cultures, and spheroids were being removed as a result. So without daily sampling, I would anticipate that all 3 vessels would have very similar expansion. We were able to obtain a total of approximately 5 billion Gibco somal iPSC cells using this expansion in the 3-liter bioreactor system. So with the cells that we obtained from the 3-liter bioreactor system, we wanted to see how we could put them to use for downstream applications. So while Michael Derr will be explaining how they can be used for differentiation, on this slide I wanted to see about cryopreserving large numbers of cells. So once we disassociated the spheroids into single cells, we cryopreserved them at high densities from 5 million cells per mil up to 40 million cells per mil. Then we placed them in liquid nitrogen over a period of time, and then removed the cryopreserved vials and thawed the cells directly into suspension cultures. So we observed that during the first passage that cells were expanding in suspension after being removed directly from cryopreservation. They did require a recovery passage where the blue bar represents the expansion in the first passage. This blue bar is significantly lower compared to what we normally see. But in subsequent passages, once the cells have recovered, in passages 2 and 3, we see high expansion similar to what we have observed on the previous slides. So this indicates that once you've scaled up to a large number of cells, you can cryopreserve them at a variety of different seating densities and then thaw them directly into suspension, only requiring a single recovery passage. And since I did mention previously that some cell lines have difficulty forming spheroids in suspension, due to this recovery passage, I wanted to see if we could improve the ability for cells to come out of cryo and then form spheroids in suspension. And by doubling the initial seeding density from our recommendation of 150,000 cells per mil to 300,000 cells per mil, we observed that more spheroids are obtained during the recovery passage, whereas the fold change remains the same. So this indicates that we're achieving more spheroids that are growing at the same rate. And when you take those spheroids at the higher density and then feed them into subsequent passages at our standard seeding density, we see again the same expansion potential compared to the previous slides as well. So this indicates that if you would like your cells to recover better, coming straight from cryopreservation, you can seed at a higher seeding density during the first passage recovery and then return to our standard density at the subsequent passages. So here are the key takeaways that I'd like you to remember. We've shown that StemScale PSC suspension medium has a simplified workflow for suspension culture, which does not require the use of microcarriers or cell strainers. We've shown that our medium is scalable across multiple culture vessel formats, from the small scale well plates up to the large scale 3-liter bioreactors. Our medium optimizes the nucleation of pluripotent stem cells into spheroids, which enables efficient growth over multiple passages in suspension. And it also maintains the pluripotency of cells that are expanded to spheroids across multiple passages. And as you'll see on the upcoming slides, Michael Derr will show how it supports downstream differentiation as 3D spheroids. So thank you very much for listening to my part of the presentation, and I would like to pass it over now to Michael Derr.
Thank you, Michael. And yes, today I will be talking about StemScale PSC suspension medium differentiation applications. And okay, so my presentation objectives is to introduce examples of suspension culture differentiation applications that we have developed, demonstrate the benefits of suspension culture differentiation compared to traditional monolayer, provide guidance and key parameters for optimizing differentiation in suspension culture, and highlight the features of PSC spheroids cultured in StemScale that are beneficial for differentiating in suspension culture. And before I get into my examples, I'd like to point out that we have several guidance documents for suspension culture differentiation applications at StemScale or thermofisher.com/StemScale, including application notes and protocols. Much of what I'm going to be presenting today can be found in much more detail within these documents. All right, so going on to the first applications I'm going to present are using PSC neural induction medium, which generates neural stem cells, and the PSC dopaminergic neuron differentiation kit, which generates specific dopaminergic neurons of the midbrain that are important for Parkinson's disease research. And these differentiation mediums were originally developed for monolayer culture, and here I optimized the protocols for differentiation in suspension culture. So just to give you an overall sort of introduction to these protocols, if we start at the schematic on top, I will kind of walk through the protocol. So first, the cells are plated in StemScale medium and cultured for 2 days to form spheres. And then next, the induction step and the expansion step are carried out in suspension culture, followed by acutase dissociation and plating the cells in monolayer for neuronal maturation. And then going down to the second schematic, the dopaminergic neuron differentiation protocol is very similar to the initial neural induction protocol. So the the specification and expansion steps are also done in suspension culture, followed by maturation in monolayer. So this strategy kind of takes advantage of the suspension culture environment and results in increased yields of NSCs and progenitor cells from these particular protocols. So the suspension culture differentiation is carried out in conditions very similar to the typical StemScale PSC culture, meaning that the agitation speeds and the medium volumes are the same for the corresponding vessels that are used, and these parameters are usually needed to be optimized for differentiation protocols, so having this guidance really saves a lot of time and kind of gives you a feel for how to do these protocols as well. And then the suspension culture benefits include shortened protocols, fewer dissociation steps, and less hands-on time compared to the monolayer protocol. So what do these protocols kind of look like during the differentiation and expansion? Here on the left, we show the neural induction protocol and kind of what the cell aggregates look like during induction and expansion. So during induction, the cell aggregates can be seen on top, and they form these sort of complex lobular-like structures that have an epithelial-like morphology. And as they continue to grow, they start to kind of break off into these smaller aggregates, which can be seen on the top left at day 6, or top right at day 6. And then this process continues during expansion, and this prevents the size of the aggregates from getting too large. And this is actually quite convenient, and is why there's only a single dissociation step during both of these protocols, and dissociation is not necessary for during induction or the expansion stages. In the middle, I have an image so you can see what the aggregates kind of look like in the 6-well plates and a 500 ml flask format. We have protocols for both the 6-well plate as well as scaling up for the larger flask formats. The 6-well plates are quite convenient for optimization experiments or for you're doing these protocols for the first time. It's a good thing to do it in smaller scale before scaling up and using large quantities of medium. And then on the right, I'm showing that, you know, instead of dissociating the cell aggregates, during expansion, these aggregates are, you know, continuously getting more cells, and the aggregates are growing and sort of blooding off. But we recommend distributing the intact aggregates, for example, 1 to 3, into multiple vessels, so that you aren't just going through medium too fast and sort of having to change the medium daily. So that's kind of how we get around the not dissociating the aggregates. And I'll go into this in a little bit more detail. Okay, so now comparing the suspension culture differentiation protocol to the monolayer protocol, by first looking at their workflows, we see that the monolayer protocol is significantly longer. We recommend going into neuronal maturation after 24 days compared to 16 days with the suspension culture. And I will show an example of why this is in a later slide. But additionally, as I briefly stated earlier, there's much more dissociation steps in the monolayer protocol. So there's 5 dissociation steps compared to only 1 for the suspension culture protocol. So right, the duration and hands-on time is decreased significantly for the suspension culture protocols. And next, we're comparing performance. So induction efficiency is quite equivalent for both of the protocols in that you achieve over 90% SOX1 positive NSCs. And then comparing expansion, the suspension culture protocol yields much more cells. So by day 12, you have twice as many NSCs, and this kind of trend continues throughout expansion as you get more and more cells. So there's equivalent induction efficiency and improved expansion in the suspension culture protocol. All right, now comparing the maturation in the suspension and monolayer. So here I look at 3 different groups of NSCs and plated them in maturation medium. So on the right, and this is why we recommend a longer monolayer protocol here. That will be illustrated. So first we have NSCs from day 16 of the monolayer protocol, and that's in the red, and the corresponding time points can be seen on the left in the red with the red star. So after 7 days of maturing these NSCs, you can see that there are a lot of flattened out cells, and these cells are proliferative. And then continuing on vertically down that column, you can see that at day 20 of maturation, these cultures result in large amounts of clumps and low neuronal purity. And then, so you can compare that condition to the middle one, which is monolayer NSCs taken from day 24 and going into maturation. And then also on the right, you have suspension culture protocol NSCs from day 16. And you see that the NSCs efficiently go into maturation, and after 20 days, they achieve highly pure neuronal populations. So to achieve the equivalent results in monolayer, more time is required. So basically, suspension culture day 16 NSCs go into maturation equivalent to day 24 NSCs of the monolayer. And that extra time is actually required to achieve that efficiency in the monolayer. Okay, so now going to the dopaminergic neuron differentiation kit, I will show an example of optimizing the size of the PSD spheroids. So looking at the protocol, you can see that we recommend culturing StemScale as PSCs for 2 days before starting differentiation. And in this example, we compared spheroids that were in StemScale for 2 days or 3 days, and put both of those into maturation or into differentiation. And the difference between these is that just that the spheroids are larger in the PSD day 3 StemScale condition. So there's a larger sphere size at the start of differentiation for the PSD day 3. And what we found is that the larger spheroids resulted in significantly lower specification efficiency, as can be seen with the decreased OTX2 expression in the ICC images in the middle. And then it's also quantified on the right. So we did quantitative ICC, looking at co-expression of FOXA2 and OTX2, and the larger sphere size starting PSD spheroids resulted in significantly decreased quantitation of the co-staining FOXA2 and OTX2. So these were not appropriately specified to floor plate progenitors. And continuing on through maturation, you can also see that this resulted in significantly lower TH-expressing FOXA2 positive midbrain neurons. So PSC sphere size at the start of differentiation is a critical parameter to optimize for differentiation protocols. And this was just an example for this one, but optimal sphere size for differentiation may vary with different protocols as well as different PSC lines. And I will show you some examples of that as well in the coming slides. So continuing with the dopaminergic neuron differentiation kits, here I am comparing the monolayer to suspension culture. And just looking at the protocol schematic at the top, you can see that, again, the suspension culture protocol requires significantly fewer days. So the time to go into maturation, the suspension culture requires 14 days, whereas the monolayer protocol requires 21 days. And again, 1 dissociation in suspension culture compared to 3 for the monolayer. And then to further compare the suspension culture and monolayer protocol performance, we compared the functional maturation of the resulting dopaminergic neurons from each protocol using 2 different PSC lines. And so to do this, we plated the cells for maturation on multi-electrode array plates from Axion Biosciences, which allows us to monitor the spontaneous functional activity and as well as the networked activity over time. And so what we found is that the neurons from the suspension culture protocol showed earlier onset of spontaneous activity and networked activity, as well as they also sustained higher levels of the activity over time. So this would indicate that the suspension culture results in more efficient maturation, similar to what the neural induction protocol showed, as well as functional maturation of these cells. Okay, so moving on to another application. So my colleague, Richard Josephson, has done a lot of work on generating cardiomyocytes from the StemScale PSC spheroids. And we have an app note online for this, as well as a protocol. And I just wanted to highlight a few key points on this slide. So the first one being, so the for this particular protocol, optimal mesodermal induction was achieved with PSC day 4 spheroids. So this is an example of a protocol requiring a different size of PSCs at the start. And the middle panel, you can see the relative gene expression from the different size of spheroids, of starting spheroids. And what he found was that using the PSC day 4 spheroids resulted in the highest level of induction of these mesodermal and cardiomyocyte markers after the induction step. So this is a good example of, depending on what protocol you're using, you may need a different size PSC spheroids. And another key point of optimization for this protocol here was it requires lower agitation speeds during maturation, because the cardiomyocyte spheroids during maturation become quite fragile. So that's another thing to keep in mind when you're sort of optimizing these suspension culture protocols, is decreasing the agitation speed is more gentle on the cells. And if you see cells breaking up, you can kind of adjust the speed to kind of compensate for that. And for these protocols, he found that beating cardiomyocytes were observed between day 7 and day 14. And the final application that I'm going to present today is the generating midbrain organoids from StemScale PSD spheroids. And again, this was done using the PSD dopaminergic neuron differentiation kit, which again was originally developed for 2D culture, and Richard optimized this for 3D culture, or for organoid generation. And this is another example of a different size of PSD spheroids being required to start the differentiation protocol. So for this one, he actually recommends using PSD day 1 spheroids. And this is really kind of a benefit of the StemScale media and its improved nucleation of these spheroids. So even by day 1 of being in StemScale, you get quite robust spheroids, so they're able to go into differentiation protocols. And this is not true for a lot of other media, suspension culture, PSD medium that we've tested. I'd also like to point out that there is a webinar available, a Lab Roots webinar by Richard Josephson called 3D Organoid Models for Differentiation of PSD-Derived Dopaminergic Neurons. So you can also check that out if you are looking for more information, and we also have a protocol for this online. And a few more details about this. So the dopaminergic neurons can be observed in these organoids by day 22, so after about 3 weeks. And then also you can see the neuromelanin staining on the right there is observed after 5 weeks. And Richard goes into how we kind of optimize this to get this expression, which is quite early compared to other examples of organoid protocols. Okay, so finally, in conclusion, suspension culture differentiation protocols were introduced for generating NSCs, midbrain dopaminergic neurons, cardiomyocyte spheroids, and midbrain organoids from StemScale PSCs. And significant benefits were seen when comparing suspension culture protocols to monolayer, including shortened protocol length, less hands-on time, and improved expansion yields. Key parameters for optimization include PSD sphere size, duration of differentiation steps, as well as agitation rates and medium volumes. And the efficient nucleation and uniform size of spheroids cultured in StemScale promote efficient and uniform differentiation, as well as flexibility during the optimization of these differentiation protocols. And again, application notes and protocols and more details can all be found online at thermofisher.com/StemScale. And that is the end of my talk. Thank you very much, Lab Roots, for having us and letting us present. And we can take as many questions as time allows. Thank you.
Thank you, gentlemen, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is, here we go. Do you keep rock when performing the medium change?
So when performing the media exchange, we only add a rock inhibitor on day 0 during seeding. So no ROCK inhibitor will be included when removing media and replacing with fresh media. You only need to add the ROCK inhibitor on day 0 during seeding.
Okay, great, thank you. And our next question, what is the effect of the spheroid dissociation on the cells? Are the cells stressed after counting?
So when we use Accutase to disassociate the spheroids, the disassociation involving Accutase is gentle on the cells, so there's no stress that will affect their viability. And we've seen that viability is affected more, like if you pass cells through a cell strainer, due to the cells essentially being sheared apart. And with Accutase, provided that you're performing the passaging step in a timely manner, there will be no issues. You can also choose to add ROCK inhibitor when you're resuspending the cells for counting to improve their health. And also, we've seen that when you scale up to large volumes, the amount of Accutase and the duration of the Accutase exposure will need to increase as well. And for 3-liter cultures, we've had them in Accutase between 20 to 25 minutes to break apart all the spheroids. And those have still been able to go into subsequent passages with no issues. So when using Accutase, it's a nice, gentle passaging reagent.
Okay, thank you, Michael. And our next question, have you tested the pluripotency of higher passages, P50 to 70, for instance?
No, we have not tested beyond 30 passages for pluripotency or expansion. We did want to provide data indicating the stability of the cells grown in StemScale over like a longer period of time, as opposed to just simply scaling up. So users can choose to maintain their cells for long-term as opposed to scaling up, but we would not recommend culturing a particular line for a significantly long period of time, similar to what you would do when treating like your adherent cultures, for example.
Okay, great. Thank you so much. And we have another question coming in. So will the recommended shaker speed, 70 RPM, work universally, or does it need to be adjusted depending on the shaker kind or culture vessel of choice?
Oh, yes. So the recommended shaker RPM of 70 RPM that I gave in this presentation is not universal and will need to be adjusted depending on a number of different variables. So if you're using the recommended, the Thermo Fisher brand orbital shaker that was in this presentation, then 70 RPM will work because that's what we use for our evaluations. But otherwise, if you have a different type of orbital shaker platform, then you will need to convert to a different RPM based on the orbital diameter of our Thermo Fisher brand orbital shaker and the orbital shaker that you are using. And we do have the orbital diameter of the Thermo shaker listed in the StemScale FAQ. So you can go look that up and perform a calculation using an equation as well listed in the FAQ. And also, if you're using like different sized flasks or well plates, then you need to balance the volume in the flask with the RPM that you're using. And so at the 70 RPM listed in this presentation, we have a number of different volumes for the different size vessels in the StemScale manual that will work. But if you want to work outside of this RPM or outside of the recommended volumes, then you'll need to do some adjusting to determine the correct balance between RPM and volume.
All right, great. Thank you so much. And thanks to the audience for so many great questions. We have a lot of amazing questions coming in. So our next question, how do you check if some differentiation appears in your culture of iPSCs?
So this can be a little more difficult with the spheroids compared to adherent culture. But I'd say for a quick assessment, looking at their morphology is a good measure because after day 1, once the spheroids have rounded up into a nice uniform morphology, if any differentiation is occurring, you'll see their morphology is actually more uneven, and it will not appear to be as perfectly spherical as all the images you've seen in this presentation.
Great. Thank you again for that amazing answer. And our next question, do you know, let's see, oh, I'm sorry, I skipped one. Can self-aggregating spheroids be transformed into organoids, greatly enhancing the understanding of disease processes, as well as aiding faster and more efficient drug testing and discoveries?
Yeah, this is Michael Derr. I'll take this one. Yes, we did show some midbrain organoid work that we've done, and indeed these StemScale PSC spheroid aggregates are compatible with organoid generation. Some, and again, there's a LabRoots webinar on that by Richard Josephson that was mentioned in the presentation. But additionally, some of our internal work, we've had good success with generating cortical neurons as well from these self-aggregating PSC spheroids. So they definitely are compatible, and those are the two that we've had success with so far.
Thank you, Michael. And we've got another question. Do you know why the starting size makes this significant difference in the DA specification efficiency?
Yeah, that's a good question. I mean, I think it could be a different, it could be a matter of sort of two different variables. Different differentiation protocols are going to require optimization of the sphere size in that particular case. It may be due to either signaling molecule penetration, so maybe the larger size decreases the penetration of the differentiation, I guess, molecules important for differentiating during specification. The other one could be sort of just the morphology of the differentiating cell aggregates during specification. So each, at least the ones that we've tested, each differentiation protocol, the morphology seems to be a little bit different during specification or during the initial differentiation process, whatever that may be. And this could have impacts on also media penetration or just how well they take on the agitation rate and other things. So it could be a morphology thing where maybe if you adjusted some of the agitation rates or the medium volumes, the larger sizes may work. So there's a few things to consider when you're optimizing the differentiation protocol.
Great, thank you again. And we've got time for a few more questions. Do you provide any training for handling 3D cultures of iPSCs?
So we do have some training videos that are available on the StemScale page. And I believe that there's also the option for a more hands-on option that can be scheduled as well. Although it might be more virtual these days.
Like everything else, right? Okay, we've got a few more questions. Can I use a passaging reagent other than StemPro Accutase?
Yes, so you can use a different passaging reagent as well if you would like to, although the results are going to be a little different. Like for example, if you try to use a clump passaging reagent like Versine, then since the spheroids aren't being broken down into single cells, there's going to be a lot more size variability when you go into your subsequent passage. So we don't recommend a clump passaging reagent for this reason. You would have to probably use a cell strainer in order to break them down further. And if you try to use another single cell passaging reagent, such as triple select, triple is actually a little more harsh than StemPro Accutase. And so this causes some nice DNA to remain in the culture when you seed your cells for a subsequent passage. And then this causes undesirable aggregation, and you get some clumps of cells instead of nice spheroidal aggregates. So if you're using triple select, then you'll probably have to add some DNA to your culture in order to prevent that from occurring. So this is why we recommend stem acutase, since it's gentle and it doesn't require any additional steps.
And I have time for one more question. Are other types of ROCK inhibitors compatible with StemScale?
Yes. So in the presentation, I mentioned we recommend using Y27632, but you can use other ROCK inhibitors. We've tested 4 or 5 different ROCK inhibitors and found that as long as you have a ROCK inhibitor present, then the cells will aggregate into spheroids. But in terms of the best performance, the Y27 has the best nucleation and results in the best size spheroids by the end of the culture period. So we would still recommend proceeding with Y27632.
Great. Thank you again, both of you, Michael Akenhead and Michael Derr. Do you have any final comments for our audience before we go?
Well, I'd like to thank you for listening to the presentation and for providing such great questions.
Yeah, same for me. Thank you for all the great questions, and thank you, LabRoots, for having us present as well. Thank you.
Well, thank you again, Michael Akenhead and Michael Derr, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.